Hello students, this is Dr. Faizan Mirza. Here I am discussing with you modifications of RNA in eukaryotes. So in eukaryotes, when the DNA template is being transcribed into an RNA molecule, which is single strand, so the RNA at that time is called the primary transcript. This primary transcript is complementary to the DNA template strand. Now, this primary transcript of RNA undergoes modification in three ways, in which eventually it will become the messenger RNA. The primary transcript undergo three modifications, including number one, addition of a guanine cap at 5' end, addition of a poly-A tail or a polyadenine chain of adenine nucleotide at the 3' end, and lastly, the removal of introns and joining of the exons called the exon splicing, gene splicing, or the RNA splicing. How this follows? Whenever you have the RNA molecule, we know that the DNA template runs from 5' as well as 3' direction. There are two strands, so the 3' to 5' strand is used as a template, and the mRNA is made 5' to 3' using the enzyme RNA polymerase. This molecule will be called primary transcript, as I have already discussed. This primary transcript will then undergo modification. At the 5' end, the guanine cap gets added and several adenine nucleotides form a tail called poly-A tail, and this gets added at the 3' end. What are the features of the guanine cap? The guanine cap and the poly-A tail, how do they function? What do they assist? They allow the messenger RNA to pass out from the nucleus through the nuclear pore to the cytoplasm. They prevent the mRNA from being broken down by the enzyme action. They prevent the mRNA molecules from joining together. So in the cytoplasm, there are more than one mRNA molecules of various proteins that are to be made. So those mRNA can join, like one mRNA can join to the other. So this guanine cap and the poly-A tail prevents that. Then this mRNA needs to align on the ribosome. So it attaches to the ribosome in such a way that the 5' end enters the ribosome first. So the first codon gets translated first. Because if the 3' end goes in the ribosome, we know the stop codon will get translated first, and that cannot occur. So these are the features regarding the guanine cap and the poly-A tail. RNA splicing, gene splicing. What are the features of gene splicing? The introns, we know they get removed. When they are removed, the exons are joined together. When they are joined together, we say they are joined up, they are attached together, and the bond that joins them together is the phosphodiester bond. The non-coding sequences, which are the introns, they have been removed, and the coding sequences, which are the exons, they are joined together, and they are the only ones which are kept. The resulting mRNA molecule is way shorter than the primary transcript that was produced. Alternative splicing can also occur. Now, when alternative splicing is occurring, exons can be rearranged in various orders. When the exons are arranged in various orders, the amino acids then can be joined in according to that codon sequence, and a different protein can be formed using the same gene in different cells or different organisms. For simplicity's sake, I have removed the guanine cap and the poly-A tail from the diagram here. I am displaying to you the RNA molecule having the exons and the introns only. At the 5' end, there will be a guanine cap not shown in the diagram, and the 3' end, there will be a poly-A tail again not shown in this diagram. This diagram entirely shows the exons and the introns. E's stand for the exons and I's stand for the introns. Now, if you look at this RNA molecule, there are 5 exons and there are 1, 2, 3, 4 introns. When the introns have been removed, how this will follow? The introns are cut and the exons are joined together. The exons, when are joined together, this will lead to the formation of a sequence in which the first exon, second, third, fourth, and fifth exons are lined in a sequence. Now, just if the cell is to make another protein out of it, they may rearrange the exons, and when they rearrange the exons, the rearrangement allows exons to be arranged in a different order. For example, the two orders that I am showing to you here are just randomly picked, in which the first and the last exon stays the same, but the second, third, and the fourth exons have been rearranged in a different order. We know that when both of these RNA or messenger RNA undergoes translation, So the ribosome will end up translating the first exon first and eventually to the last exon, which is fifth. These blocks represent the amino acids. So the same amino acids will be joined but in a different order. So when the same amino acids are joined in a different order, you end up having different proteins being made because of alternate splicing. So this is what alternative splicing will do. It will allow different proteins to be produced using the same gene. Moving on to the role of telomeres. We know that telomeres are present at the end of chromatids. The chromatids, the chromosomes in eukaryotes, they are the linear DNA, and the linear DNA ends are exposed. They are vulnerable to getting enzymatically cut, or they can undergo changes, modifications. So the telomeres are there to prevent all this. So they are present at the ends of the linear DNA of chromatids and chromosomes. They prevent the loss of genes, which is the coding sequences. They prevent the degradation of the ends of the DNA from the enzyme action. So the enzymes do not recognize the telomere to be removed. These telomeres, they also prevent the ends of the chromosomes from fusing with one another. For example, chromosome number 1 and the chromosome number 5 in a cell, they may be together, close together. Their ends, they do not join up. They stay separate chromosomes. Why? Because they both have telomere each that stay separated. Then these telomeres, they allow the DNA replication to continue, because when the telomeres keep shrinking, so more cell cycles can continue until the telomeres are shrunk altogether. They allow some cells, specifically the stem cells, to carry out repeated cell cycles continuously. Repeated mitotic cell cycles continue because the telomeres stay lengthened or elongated. They also prevent the end of chromosome from being recognized as damaged ends. So these are the two typical topics that students at time find a bit tricky, like what exactly is RNA modification, which occurs in eukaryotes, and the role of telomeres. So I've tried to cover this in this video. I hope this was helpful for you. That's it from my side. Thank you so much.